Good morning, everyone. I'm Theresa Currier Thomas. I'm an associate professor um, and studying or over here at the Phoenix or at the I'm sorry, Phoenix Biomedical Campus on the UA Comp campus. Um, my research is predominantly looking at the etiology of late onset deficits caused by traumatic brain injury with a focus on evolving pathology that occurs even after a single injury. And so um, today I'm going to talk to you a little bit about some of our early rehabilitation paradigms that we've been using and kind of introduce you to some of this invisible aftermath of what's going on pathologically in the brain. I have nothing to disclose except for the fact that I like to make fun art out of my histology images. So this is I don't know if you can see can you see my cursor? Yes, no?
Yeah, we could see your cursor.
Yes.
Okay, so this is a ramified or surveilling microglia, and then these are all types of different activated microglia that we found in the brain after traumatic brain injury. So that's my fun with histology. So I also want to take a quick moment to start and acknowledge my lab. I have an incredible group of dedicated and talented scientists who are at the heart of everything we do. For those of you who aren't familiar with preclinical research, especially the kind that we do in our lab, it requires an enormous amount of time, technical precision, and lots of patience. And these folks consistently show up with curiosity and resilience and like a deep commitment to study the science. And so they're truly my second family. Or if you ask my husband, maybe my first family, since I spend so much time with them. But I'm so proud of the science we've done together and the spirit in which we've done it. So the objectives are that we're going to look at our preclinical model of traumatic brain injury and look at its value for studying mechanisms of prevention and treatment strategies for late onset symptoms, specifically looking at some maladaptive circuit reorganization and efficacy of early circuit-directed rehabilitation. And just for funsies, I'm going to add in some little tidbits regarding what we've needed to do to include sex as a biological variable in these experiments, because it is rather unique. And so according to the CDC, approximately 3 million people a year in the US, 69 million worldwide visit emergency departments for traumatic brain injury. 90% of those are mild traumatic brain injury or diffuse or concussions. Nearly half of these patients do not return to their pre-injury level of health or well-being by 6 months post-injury. And despite knowing this, there are no prophylactic treatments that currently exist, especially or specifically for mild traumatic brain injury, but for traumatic brain injury overall. And there's very little that has been able to prevent or mitigate these persisting or late onset symptoms. So mild traumatic brain injury accounts for the largest proportion of an $80 billion annual economic burden in the US. So even if only a small percentage of these mild traumatic brain injuries reduce or result in long-term problems, the sheer volume of cases drive up the total costs. And some of the persisting late onset symptoms are potentially or likely effective. They require repeated medical care, rehabilitation, that the 80 billion involves time off of school or for adults work, decreased productivity, and disability and mental health claims. Children and adolescents are the second most vulnerable group, with about 1 to 2 million pediatric mild traumatic brain injuries reported annually. This younger population has a lower mortality rate than the number one most vulnerable group is over 65. Yet they face significantly increased risks for mental health, psychiatric hospitalization, self-harm compared to peers with orthopedic injuries. And so moreover, due to their age, the persistent symptoms tend to last longer and may interfere with critical neurodevelopmental or educational milestones. So a persistent post-concussive symptom is considered persistent when it lasts beyond 3 months. And late onset is when it evolves after the initial symptoms have have uh, resolved. And so and then there's different patient trajectories that are also associated with this, with some patients that will decline over time, some will worsen over time, and others will experience, again, these late onset symptoms. These symptoms generally fall into three broad domains: cognitive, somatic, and affective. And notably, the majority of patients experience affective symptoms. And just a little note, in case you're interested, is that up to 50% of traumatic brain injuries will report some type of affective symptom or increased affective symptom or increased severity in affective symptoms within the first year post-injury. This is similar for both stroke and cardiac arrest. And despite the commonality, we still lack a mechanistic understanding of why affective symptoms are so prevalent. So females are typically have been underrepresented in academic research involving traumatic brain injury, mostly because emergency department reports tend to be 2/3 to 3/4 males. And then preclinically, everybody's a little bit afraid of dealing with the estrus cycles and don't understand how the estrus cycles can influence the outcomes. But it's becoming more and more prevalent, the need to study females. And especially with the studies that are coming out now with domestic violence, where TBI is impacting up to 20 million people a year via domestic violence. 3/4 of those are women, and many of the other ones are children. And this is greater than the amount of emergency department reports and military combined. Our cycling women have more symptoms, greater duration. They report menstrual problems, lack of a cycle or irregular cycles. And when I published this paper back in 2022, only 7% of preclinical TBI research included both sexes. So when we talk about rehabilitation, I wanted to go over a little bit about what is typical for an emergency department discharge. So after a kiddo is seen for concussion and is deemed an uncomplicated concussion, so there's no CT, no evidence of bleeds, they're typically counseled before going home that if the symptoms worsen within the first 24 hours, they should seek immediate care. They should restrict their cognitive or have restricted cognitive and physical rest for 48 to 72 hours, avoid screens, reading, work, and loud environments, no exertion, et cetera, et cetera. Once they start feeling better, gradual return to activities, as long as they don't exacerbate the symptoms. And this is not for sports. Sports is handled very differently. But there are some major gaps. And some of those are, there are no gaps for patient education on potential for persisting symptoms, any kind of efficacy or any kind of things that they can do to prevent those late onset. There's a lot of time there's no support network education to let the children know, or to let the people that support the children know of the symptoms and kind of what they're going to be going through over the next several weeks or months. And any kind of reactive care after they're discharged or any other kind of care that they get is reactive and not preventative. And so often with moderate traumatic brain injury or stroke, they'll send them home with exercises that can promote thalamocortical structural activation, but that doesn't happen for mild traumatic brain injury. And I often like to talk about or to introduce this as you know, our brain injury patients, they have no scars, they have no casts, they look perfectly normal. A lot of them, especially if they're playing sports, they revel in it, that they enjoy that interaction. But they also know that they feel off. They're often exhausted, forgetful. They may feel overwhelmed, and they feel, and not only do they feel different, but their friends and the people that know them well notice they're different, but they don't understand what they can't see. And then often it gets explained away as puberty or an attitude problem in adolescence. And so a lot of times they think it's all in their head and you just need to suck it up or figure it out. And so there's not that patience associated with it. And so when concussions occur, or moderate to severe concussions, we often, so a diffuse axonal or a diffuse traumatic brain injury in general results from the mechanical forces that cause rapid acceleration, deceleration of the brain. And it produces, depending on severity, widespread axonal injury without requiring a focal impact. And so the hallmark pathology for traumatic brain injury typically is diffuse axonal injury. And what it does is it happens within these white matter circuits, and it causes a disruption of circuit function rather than a focal lesion. And then it can also occur, it can occur with overt lesions or without. So it can be as part of a more severe injury or as part of a mild to moderate brain injury. So how do we look at diffuse axonal injury in our animal models? So we have a midline fluid percussion injury device, and let me see. So we anesthetize or we do a midline craniectomy, midline between bregma and lambda. We affix a female lure lock into that craniectomy and seal it under anesthesia. And then we attach them to this fluid percussion device, the cylinder, and we wait for the animal to awake. And so we'll gently pinch its toe until we get a pedal reflex. And then we drop the pendulum onto the cylinder and get a fluid pulse. And that gives about an atmospheric fluid pulse of about 1.9 to 2.1 in males. And what we get is a fencing response. And so up here you can see in our rats, this is before we drop the pendulum, and this is up to 17 seconds after we drop the pendulum. And so you can see that the fists are clenched in a tonic posturing position, similar to what we see often in football, MMA, hockey, and several other sporting incidents. And then our animals have a loss of consciousness. We can't say consciousness, so it's called writing. It's a writing reflex time. So we put them in a supine position and we wait for them to flip over. And that usually for all of our studies, we get 100% of the fencing response, and we get about a 5 to 12 minute writing reflex response. Now, what's interesting is when we added females into these and we connected them and did our first injury, our females at the same level as males had longer writing reflex times, would have seizures, and had we had increased death resulting from the fluid percussion injury, which required us to optimize it and lower the injury fluid pulse to include them in our experiments. So what does diffuse axonal injury look like in our rats? So on the left, you can see diffuse axonal injury in a human patient, and here in the thalamus of one of our rodents. And so this is an amyloid precursor protein stain. These prominent bulb-like structures under the arrow represent axonal varicosities, or sites where the axons have undergone this mechanical shearing due to the diffuse axonal injury. And then following that transection, the axonal membrane rapidly reseals, but the intergrade axonal transport is disrupted, leading to this focal accumulation of APP. So that's what we're looking at here in both the thalamus of the human and the rat. And these injured axons, the point I want to make is that these injured axons and injured neurons are adjacent to healthy neurons. And they don't necessarily die, and I'll show you sometimes that they can actually start to try to reconnect. And what happens to that axonomized axon, or we end up instigating this chronic neurodegeneration. So this is silver stain, or dark aminocupric silver stain. And so that dark agrophilic deposits that you see throughout You can see all the way starting at 1 day post-injury, all the way out to 28 days post-injury. So revealing a progressive neurodegeneration in the weeks following our fluid percussion injury. And so this ongoing structural disruption is thought to promote adaptive and maladaptive neuroplasticity and contribute to the late emerging sensory or late emerging impairments that we may see. So due to the axonomy, we wanted to look more at the thalamic axons and neurons because we thought that maybe because of the axonomy, they'd be uncoupled from the white matter tracks. So we use Golgi staining, which randomly and indiscriminately labels neurons, and that allows us to do a 3D reconstruction of these processes. And so at 7 days post-injury, here's our neuron. You can see that we have fewer branch points, endpoints, process length, and then we also have an increase in soma area, and this could reflect an enhancement in transcriptional and translational activity because when we looked at 28 days post-injury, those processes were now similar to our sham. But if you look closer at our complexity and look at the 3D reconstructions, you can see an even distribution of these processes in our sham animals. However, at this 28-day, and we're looking at like the structural complexity, you can see that the distribution is much less normalized, and it's indicative of perhaps that they're not reconnecting in the same location, and perhaps indicative of what could be causing hyperconnectivity. And so this brings us to our working model of how late onset symptoms can occur, and that once a circuit is disrupted, the late onset morbidity emerges during the brain's attempt to recover. And so what happens between the injury and the repair are a lot of different things that are going on that gives us that requires a time course for us really to understand the dynamic processes leading towards morbidity and the need for a simpler circuit so that we can demonstrate these features. So we have, amazingly, identified a simpler circuit that demonstrates late onset morbidity. And we found this in the whisker circuit. And so this was an observation that when the shams were stimulated, you can see that they kind of ignore the stimulation, they actively rear and explore, their tails and bellies are lifted. However, in our injured animals, you see that he's flat to the ground. They're freezing. He's actively avoiding the stimulation of the whiskers. And so we've got several different things that we look at that we can count. And at 7 days post-injury, this isn't apparent. But at 28 days post-injury, you only see this when they get this constant stimulation. And so, so this gives us a circuit that we can focus on to look at and study late onset symptoms. So why is it, what are the benefits of using the whisker barrel circuit to study late onset symptoms and circuit disruption and neuroplasticity? For one, it's our rat's primary sensory modality. So it's similar to vision and hearing in patients. And so, but even more cool is, and you'll never look at your rats or cats or dogs again the same, but every one of the whiskers corresponds to discrete locations within the brain that allows us to map circuit function and integrity. And so the sensation of the whiskers translates through the VPM of the thalamus and then into the cortex. And here is another depiction of it from a glutamatergic relay from the midbrain to the thalamus, from the thalamus to the cortex. And so this circuit has fundamentally, is very simplified in that it's glutamatergic, except for GABA innervation from the thalamic reticular nucleus, and then a little bit of norepinephrine from the locus ceruleus. And so this provides us with a behaviorally relevant circuit that is far less complicated than cognitive and emotional circuits, and allows us to study mechanisms mediating circuit function. And then hopefully we can, and then as I'll show you at the end, extend what we've learned back to those more complex circuits. So once sensory hypersensitivity develops by 1 month post-injury, it persists to at least 2 months post-injury. It occurs in both sexes. So here's the whisker nuisance scores for the males and the females. However, in the females, males tend to have more severe symptoms than females. However, in both, the severity of the whisker score correlates with evoked glutamate release in the thalamus, not the cortex. And so this thalamus-specific glutamate accumulation is why many of our studies now focus on the thalamus as the key node of circuit dysfunction. And so clinical care for post-concussive symptoms is more likely, again, to happen after the symptoms develop. However, there are a constellation of symptoms, and these symptoms are diverse, but one commonality of them is that a lot of them have a thalamocortical circuit aspect to them. And so if we can provide evidence that we can improve circuit integrity and prevent or lessen chronic or late onset symptoms, perhaps we can get early rehabilitation integrated more into the standard of care. And so our hypothesis is that early rehabilitation will promote adaptive neuroplasticity, thus preventing or reducing the severity of late onset morbidity and associated neurotransmission. And so that's kind of here where we think we show that there's a loss of circuit integrity by the smaller bars, and then increased connections by new growth. And so we believe that rehab it's not going to do anything with these new synapses, but it's going to strengthen that original circuit. And so how do we do this? So we developed a circuit-directed rehabilitation strategy focused on the whisker circuit. Most preclinical rehab studies use global approaches like environmental enrichment or exercise, which make it difficult to isolate causal effects. And so our approach uses tunnel therapy, which is a task specifically designed to engage the whisker circuit. And we found that 15 minutes of tunnel training upregulated BDNF expression up to 5 hours gene expression. And so when we apply it 3 times per week for 2 weeks, we also found that it could reduce whisker sensitivity in males. And so we then wanted to integrate it into a full study. And so, and we also wanted to integrate females into the study. And so just a little bit of what we had to pay attention to with the females is the pheromones, especially with animals. So we had to make sure that our males and females were not in mixed cohorts, that their pheromones didn't get a chance to interact because it would disrupt any kind of behavior, which included thorough cleaning. But we also started tracking estrus cycles. And so a rat's estrus cycle is about 4 to 5 days long. And so, so we started it a week in advance so that we could track to the day that we injured. And then we also had to control that. So we were handling our males every day. We had to handle our females as well. Handling our females every day, we needed to handle our males too. So they were controlled by doing a gentle swab to the anus. And so we also had to make changes due to the body size. The females are about 30% smaller than than males. And so when we do microelectrode arrays to measure in the thalamus and the cortex, we had to ensure and come up with new stereotaxic coordinates to ensure we were in the right location. So for these studies, we injured at the first day, and at 1 week post-injury, we started rehab. So during week 2 and 3, and then at 28 days post-injury, we looked at the whisker nuisance task, and then we did electrochemistry. And so what we found here is that our light gray are our shams, that rehab didn't have any significant effect of our shams in male or female, but we did get our significant increase in the whisker nuisance task scores, and that early rehab was able to reduce those by 40%. But what is more striking is that we typically see evoked glutamate release in our males and females post-injury at 1 month. However, this early rehabilitation not only improved the symptoms, but it changed the circuit function underlying it. And so overall, this supports that early rehab improves symptoms and normalizes underlying thalamic neurotransmission. What's interesting is, again, we show that the females have this overall decrease, but when we look at the cortex in males, we also see an increased evoked release of glutamate, which we don't see in females. So the fact that we're seeing this normalization or prevention of changes in glutamate neurotransmission in the thalamus in both is remarkable. So for those of you who aren't as familiar with the thalamus, it acts as a central relay station for sensory and motor signaling, and deficits and too much glutamate, potentially hyperconnectivity within this area can span to other nuclei, thalamic nuclei, therefore potentially causing deficits in regulation of consciousness and alertness, cognitive and emotional processes, as well as the sensory and motor modalities. And so, these apologies. So the central relay hub is then broadly impacted, and it also suggests thalamic vulnerability in long-term TBI pathology, where a lot of studies focus on the cortex, and it defines a concrete behaviorally relevant target that for our interventions and recovery tracking. And so what we also found in this study was that TBI impacted the estrus cycle in our females. So our females ended up spending more time in diestrus and less time in estrus over a 28-day period. So it wasn't an acute function, and that's why I have these graphs out here, is that this was prolonged over the 4 weeks post-injury. We also noted that regardless of injury, our females had slower glutamate uptake rates and clearance. And so what does this mean is that typically if we locally apply glutamate, it's very quickly removed from the extracellular space in rats. But it takes about an extra second for it to clear in females, meaning that any kind of drug that's modulating glutamate neurotransmission may potentially interact with the system a little differently. And so the take home from these studies is that early rehabilitation can prevent or reduce the emergence of late onset symptoms, that the thalamus is the central hub for the chronic symptoms, and that males and females show distinct neurobiological responses. And so with this, one of the things we need to do is look at later time points to make sure that that prevention of 10 days between us stopping rehab and looking at the animal, if we took that out to another month, is that a prolonged change? And is the change in the neurotransmission prolonged? And for those, I apologize, a month post-injury is about a year post-injury equivalent clinically. And so some of the significance of this is it demonstrates that early rehabilitation can disrupt the progression involved with chronic symptoms, and perhaps shifting that therapeutic focus towards the thalamus can really drive the types of rehabilitation that are advised, and that the sex-specific circuit responses indicate the necessity for including sex as a biological variable in preclinical trials, as well as looking at it in clinical trials. It supports circuit-directed rehab as a scalable, translatable strategy for studying the impact of rehabilitation, and establishes a model for when and where so that we can identify when and where interventions are most effective. And so one of the things that I showed you was the hyperconnectivity or the increased glutamate in the thalamus. And so we wanted to look at what was potentially causing that. And one of the molecules that we've identified that's specifically upregulated in the thalamus post-injury is thrombospondin 1. And so thrombospondin 1 is a low-expressing astrocyte-derived molecule that is it's involved in neurodevelopment and then downregulated before the age of 2 clinically or 21 days in our animal models. And then we find that it's upregulated this first week post-injury. And thrombosponin works by binding alpha-2 delta-1 calcium channel that is implicated in triggering epileptiform cortical activity. However, it's never been studied after diffuse axonal injury, and it's not been looked at in the thalamus or in its terms for its involvement in long-term effects. And so gabapentin can allosterically block this thrombosponin alpha-2 delta-1 binding. And because through allosterically blocking it. And so we wanted to give gabapentin for 1 week post-injury. And what we found is that without gabapentin, with just saline, we saw an increase in co-localization of synapses. So green is presynaptic for V-glute 1, 2, and red is postsynaptic for PSD95. And then the yellow is this co-localization indicating a active puncta. And so we see an increase after injury, and then we see that gabapentin was able to prevent that. So we went on to give gabapentin for the first week post-injury and look at late onset sensory hypersensitivity. And what we found in the whisker nuisance task was that we saw the significant increase in our whisker nuisance scores, but we did see, at all 3 levels of gabapentin, 3 doses, we saw a decrease in severity of symptoms. So this is an indication that, again, something that we can give early can have a long-term impact. And so our goal for the upcoming studies are to combine this. So use rehab to drive neuroplasticity and strengthen connection, and then use gabapentin to reduce maladaptive synaptogenesis such that we can have hopefully no morbidity or very mild morbidity. So some of the other things that our lab is working on is looking at how diffuse axonal, I've showed you that diffuse axonal injury can cause persisting neurodegeneration out to 1 month post-injury, but what's going on longer? And so we wanted to look at different pathological outcomes. And we also wanted to introduce this pathology into include females. And so here we looked at GFAP, glial fibrillary acidic protein, which when upregulated is indicative of astrogliosis. And what we found, so here are our sham animals, and we looked at 7, 56, and 168 days post-injury. These are our male sham and injured, and then here are females. And so what we see at 7 days post-injury is an increase in GFAP in males and females. But what we see in our shams is that there's an increase over time, such that in the thalamus, it's not that GFAP is going down and gliosis is going down, it's that the aging of our animals is causing gliosis. And that's very common or is very well documented in the literature. We also looked at the shams and we looked at the cell counts of the astrocytes, because it's one thing to have more astrocytes, it's another thing to have, but so having more astrocytes and having more GFAP aren't necessarily 1-on-1. And so what we also found here out to 56 days post-injury and still extending would be significant if it weren't for the changes in our shams, is an increase in the number of astrocytes as well. We also looked at microglial activation using IBA1 staining as an indication of neuroinflammation. And so here is a ramified or non-active microglia. And then here you can see some phagocytotic and bushy and amoeboid types of pathology. And so normally microglia are evenly distributed within the tissue, and so they don't tend to overlap. You can see even more clearly here in females is that they all have their own area that they're surveying. But when there's pathology, when pathology occurs, they turn into this amoeboid form and they go through to phagocytose, all that degeneration. And so you'll see an increase in the number, which we measured here in the soma count. But when they become activated, they also retract their processes and have fewer endpoints. And so we looked at these over at the 756 and 168 day time point. And in males, we see a significant increase in the number of cells. This is significantly greater than what we see acutely in females. By 6 months post-injury, our males are similar to the sham controls. However, our females are still elevated. And this is also shown with shorter branch lengths is what's being shown in these and the number of endpoints. And then one of our students went through and characterized and profiled all of these and found that these changes were most predominantly driven by the amoeboid form. So what that basically means when you have the amoeboid form is that there's active neuroinflammation, active pro-inflammatory cytokines, and that these are ongoing in our thalamus chronically, where they can evoke more havoc and potentially promote a more neurodegeneration and more cell death. And so we wanted to look at the Wallerian degeneration. So this is a silver staining that I showed you before. And we wanted to look at it throughout all three regions. So in the thalamus, the cortex, and the reticular nucleus. So I'd shown you this diagram earlier. So here in the thalamus, we see, again, the both male and female degeneration for out to 6 months post-injury. In the cortex, it tends to decrease over time and is there's still a little bit present, but it's not as robust. But we also found this chronic neurodegeneration in the reticular nucleus as well. And so the reticular nucleus, the thalamic reticular nucleus is important because it's the primary, it's not only the primary GABAergic modulator for the VPM, it's for the thalamus. And again, our initial, in a normal thalamus, when we saw the increase in glutamate, we thought we were seeing, okay, well, we're getting regular inhibitory, and now we're seeing, and it's just an increase in the glutamate release in the thalamocortical circuit. However, now with this increased neurodegeneration in our reticular thalamus, we also believe that there is a lack of GABAergic innervation into the thalamus that's promoting these late onset symptoms. And so we now really want to start targeting and looking more at what our effects are on the reticular nucleus with a rehab or any kind of pharmacological innervation, as well as looking at how it impacts other thalamic nuclei. And so the thalamic reticular nucleus is composed entirely of GABAergic inhibitory neurons, and its primary role is to provide inhibitory gating of excitatory thalamocortical relays. And it regulates the sensory input, attention, arousal, cognitive function, sleep rhythms, all of these that are implicated as chronic symptoms after traumatic brain injury. It also filters ascending sensory information before it reaches the cortex. So it's essential for that attention modulation, so preventing that sensory overload. And so even subtle disruptions in reticular thalamic nuclei function can produce widespread network effects, particularly on sensory gating and attention. And so we want to, in order to study this, we've been taking our microelectrodes that we've used for our glutamate, and we've been transitioning those to measure both glutamate and GABA. And so here is one of our microelectrodes. These are about 333 microns long and about 15 microns wide. And we go in and we can hand coat them to make them selective for glutamate in our previous experiments. But in this case, we did glutamate and GABA. Such that we could record both of them and look at the excitatory inhibitory ratio. And so this is a calibration of that. And then this is where we put the electrode into the cortex. So the blue is glutamate and the black is GABA. So you can see that we have a 2-to-1 ratio of glutamate to GABA here in the cortex. And then you go into the reticular nucleus, which has projections from the cortex and other areas to regulate it that are glutamatergic and GABA. So you see a much higher GABA, 1-to-1 GABA tone, glutamate GABA tone. And so, and so if we go through this, we're working to put these into the freely moving animals such that we could stimulate their whiskers and see how this is affecting their awake behaving and their excitatory inhibitory profile while moving. And we did some tail pinch studies also to look at male versus female. And so we get these little micro spikes when we do a tail pinch. And we see that some of this, awake behaving electrochemistry has never been done in females before. So these are the first ones that have ever looked at male-female differences. And so again, we looked at these little micro peaks and found that they're significantly more in males. Well, it's not significant, there's an N of 2, but so far we're finding that there are more micro peaks in males than females. So as I mentioned, this pathology is not just specific to the whisker circuit. We also see changes in other regions of the brain that we can measure. So here we did an open field test with these animals at 1 week and 1 month post-injury. So they had similar distance traveled, but by 28 days post-injury or 1 month, it became significant that they spent significantly less time in the center of the open field, and they had fewer entries. And when we looked at signaling, we put our microelectrode into 2 nuclei of the amygdala, the BLA and the CEA. And we found that the CEA, not the BLA, is the one that showed most of the changes in glutamate neurotransmission, with a decrease in evoked glutamate release and slower clearance rate. And so why this is important is that the CEA is the primary regulator of the hypothalamic pituitary adrenal axis. And so we'll go directly to the hypothalamus to regulate cortisol release. And so this transitions into one of the other passions that I've come around to bringing more education to is the neuroendocrine system after TBI. And so it's becoming more and more, while much of the work focuses on that circuit reorganization component, we've identified evidence of hypothalamic pituitary adrenal axis dysregulation early in my career, and I've been working with physicians and scientists alike to increase awareness and assist in developing better clinical models. But coincidentally or not, I just wanted to point out here are chronic TBI symptoms, and I color-coded them. And I know, I'm sorry they don't show up as well on here, but for growth hormone deficits, cortisol deficits, thyroid, and testosterone, they're color-coded to where many of these are part of the chronic TBI symptoms. And so it's really important for us to start to bridge that gap with neuroendocrinology when treating our chronic TBI patients. And I wanted to do a case study that we published here from Phoenix Children's Hospital, which was a 15-month-old boy presented with failure to thrive. He had a history of having a traumatic brain injury at 6 months old. And his height and weight, by 13 months old, his height and weight were both below 10%. But it wasn't until he was 15 months old that they ran the hormone panel, and he had a low T4, and he had undetectable IGF-1 and IGF-1 binding factor indicative of growth hormone deficits, and then he had a normal cortisol test. And so he went on a clinical course of treatment, and then within 33 months was at the 25th percentile for height and weight, but at 7 years old then developed central adrenal insufficiency. And so what I really wanted to point out here is that it is something that we see in our pediatric population. and it emerges over time. So it took it 15 months to emerge, and then at 7 months or 7 years old, we're still seeing deficits. And so it also requires this ongoing endocrine surveillance. But what I think is very exciting about it is that this development over time gives us, again, another opportunity to intervene with interventions. And so it's why we need better preclinical models so that we can understand the trajectory and what's contributing to this long-term compromise of the hypothalamic pituitary axis. And looking at our models, again, I mentioned that we study a lot of the hypothalamic pituitary adrenal axis. So immediately after our injury for 24 hours, we get increased cortisol levels. By 2 months post-injury, those cortisol levels are decreased by 60%. And then when we put them under stress at 2 months post-injury, we get a blunted stress response. And this data has been published, but some of our newer data looking at more acute time courses, this is a glucocorticoid receptor, which is the primary receptor for cortisol or corticosterone levels in rats. And so we can see dynamic changes at 1, 3, 5, 7, and 56 days post-injury. This is looking in the amygdala, where we can see a decrease. And this decrease in glucocorticoid receptor makes sense because when cortisol is high, the glucocorticoid receptor is going to downregulate so it doesn't get overstimulated. So it can cause negative feedback towards that HPA axis. However, when cortisol gets low, it's going to increase to ensure that it's able to sense and react appropriately. And so that's where, again, we're seeing an increase in these glucocorticoid receptors. And so we kind of put all this data together, and what it looks like we're seeing is that it's not this chronic deficit, at least for our injury model, that we're seeing, that it's cyclic, that we're getting too much cort, and then we're getting hyperreactivity. And so it's potentially cyclic, which has which suggests kind of this paradigm shift of how we understand and how we can treat post-TBI symptoms. What is also interesting is that when glucocorticoid receptors are up, BDNF, which is a neuroplasticity receptor, also goes up. And when it goes down, it tends to go down. And so this shows an interrelation between what's going on in our stress system and what's going on with neuroplasticity that's potentially being driven by our or modulated by our rehab, and something that we need to take into consideration as to whether our rehab is effective or not. And so, again, overall, this is potential mechanisms for increased risk for affective disorders after TBI could influence our rehab. And then changes in cortisol because of the glucocorticoid receptor being directly on glutamatergic and GABAergic neurons can influence directly and indirectly glutamate and GABA neurotransmission. So it's complicated, right? We also looked at about 6 weeks post-injury. We wanted to look at circulating levels of growth hormone. So we looked at plasma levels, and we see an overall like a trend towards an increase in growth hormone, which is not what we saw clinically. But what's interesting is when this was done in juvenile rats, so rather than 8-week-old rats, 2.5-week-old rats, they're seeing this dichotomy as well as some very high scoring and very low scoring or levels of growth hormone. We did see a significant decrease in thyroid stimulating hormone, but only in males. We also saw a significant decrease in luteinizing hormone, but only in males. And then we did see this interaction here with prolactin. And together, what these data are suggesting to me is that that the sex-specific pattern suggests a disinhibition of the hypothalamus. Again, looking at some of those GABAergic innervations, where selective failure of releasing hormone circuits or loss of inhibitory input, particularly, either from dopamine or towards somatostatin, could be contributing factors. So in closing, thank you for your attention. But I do want to say often when patients are told it's all in your head, they're right, but not in the way that the people actually meant. And so this is just showing you different levels of thalamic neurodegeneration, microglial activation, and astrocyte activation. And with that, I will take any questions.
Theresa, this is fantastic. Thank you so much for coming this morning and sharing your work.
Thanks, Jason.
Theresa, what do you think it's going to take to get clinicians to essentially change what's done with an eye for preventing the long-term sequela? Because typically, correct me if I'm wrong, typically patients aren't followed long-term unless they have other symptoms.
Right, and that's the problem that we're trying to address. Again, the discharge is typically, the symptoms will resolve, and they're not even told about long-term or late onset symptoms occurring. And I do think we have several issues that we have to overcome. I mean, first is insurance. If there's no justification for it and no proof that it's going to make an impact, why would insurance approve it? So I think, and then it's also, I think, better understanding of the circuit changes that occur after brain injury. I mean, when we start engaging the thalamus and different types, a lot of rehabs, therapies and dual therapies already address some of the paradigms, but now that we're able to more specifically model it, we can show how it's changing neuroplasticity. So I think that it's just evidence is needed, and then and showing it in our preclinical models, hopefully giving that and the impotence of talking to clinicians and seeing if that's something that they can start to do is to recommend different types of small strategies and work with us to come up with some kind of paradigm so that we can send everyone home with a kind of a post TBI, what you need to know flyer or handout or booklet.
Have there been studies that actually follow patients long-term? Long-term meaning essentially equivalent to what you've shown in the rats or even longer than that to show that there are indeed some of these long-term sequela that people just don't even realize happen?
Yeah, so there are many studies that have followed patients out to 10 plus years post-injury. And that's where we come up with a lot of the statistics of anywhere from 10 to 50% of those patients present with either late onset or persisting symptoms. And again, with the late onset, it's very difficult to track that back to the traumatic brain injury itself because often TBI isn't a question that is in the patient history. But it's also because it's the trajectory of the neuroendocrine is long. So when we're looking at 6 months post-injury, it's like 10 years post-injury clinically. So it can, have a, so making that connection and then having someone look for neuroendocrine disruption when there's no evidence that there should be, a lot of it is education. I think when it comes down to it, it's education and it's implementation of things that are small to kind of track our populations longitudinally. And with the pediatric population, it's even harder because as they mature into adulthood and they go into adult care, a lot of that continuity between rehabilitation and treatment is lost.
Are there any more questions for Dr. Thomas?
Actually, Theresa, for our trainees on the call, can you just give them a bit of an idea of how many years of work this has taken for you to get to where you are, what the foundation's been, sort of how you've gotten to where you've gotten to, and what kind of, things young physician scientists should be thinking about, if they want to embark on a research question like the one you've asked?
Oh, that's a great question. So I've been in neuroscience research for 25 years. The rehab paradigm, the late onset sensory, and again, everybody's like, oh, whiskers, but it's actually very special because it's one of those, it's one of very few reproducible late onset symptoms. So we've actually reproduced it in over 8 publications. It's now in different labs. So the ability for us to track um, neuroplasticity and the impact of rehab through that is incredibly powerful. Um, some of these studies, and I'll flip back here. I've been working on this with the HPA axis for over 12 years because we first thought that it was just going to be this decrease in cord, like we see in other deficiencies, and it's not a deficiency at all. It's a dysregulation, and so again, coming to that consideration of it's whether it's a dysregulation or a deficiency may be injury severity dependent. See, this microelectrode work, again, these being the first going in, the person who actually created the microelectrodes was my PhD mentor. So I've been working with this for over 25 years, and this would be the first implementation of these glutamate GABA electrodes. So what does this have to do for physicians and kind of, where it goes is that there are powerful connections when you see something in the clinic that we as experienced scientists can replicate in the lab and really piece it apart. But it's also when you're going to treat, potential symptoms and you want to know what the long-term consequences are. It's one of the things that we don't do in science or medicine even close enough is look at the long-term consequences of what we do acutely. And so, yeah, it makes our patient healthier, but is it going to make them better in the long term, or do we even know? And so I think those longitudinal studies clinically are very important and tracking those. And then a lot of them to look at sex differences usually don't have enough power. And again, that's often because it's more males than females that present in the emergency department. But yeah, all of this is an accumulation of many, many years of work. And I kind of glossed over it. But there are but it's also giving us this whole network of looking at the entire circuit and associated circuits that we can really evaluate the impact of whether it's rehabilitation or early interventions. Jason, does that?
Yeah, no, that was great, Theresa. So, for our trainees on the call, the value of having partners like Theresa is that, as physicians, you guys know a lot of the relevant questions that need to be asked. You know what's affecting your patients. Maybe you don't know why. And there are a lot of reasons why trying to understand the why in clinical medicine is incredibly difficult, and you need to bring it back to the laboratory. And so having partners like Dr. Currier is what enables you as a clinician to identify a relevant question, something that you think would greatly impact patients or something that you think would greatly impact our understanding of disease. And then you take it back to Dr. Currier and you say, how do we do this in a systematic, methodologic way in the lab where you can manipulate conditions that you could never manipulate in a human? You can't obviously cause a TBI in a human. You can't turn genes on and off in a human. And you can't even do it nearly on the time frame that Dr. Currier has showed you. I mean, trying to study pediatric to adult disease would take decades in a human, whereas it is a much shorter time frame in an experimental animal. And so to me, when I listen to these talks, I really challenge you guys to think about how could you apply this in your career and make it a meaningful academic endeavor. So thank you, Dr. Currier. I think it's really, really helpful for everyone here, myself included, to see how you sort of you did this over a career arc. And I think that's really important. Thank you.
Thank you. I was just going to say, just quick before everybody goes off, that the rats are faster. They're at accelerated aging paradigm, but we can also move some of these into flies that we've been working on for the last year. And so this is, we put the flies into a little container, and then we injure them. So hopefully this isn't too loud. But I wanted to point out that you can see their wings are splayed. And this is actually modulated by the same area of the brain that does the fencing response. And so these allow us to do like aging studies in 15, 20 days rather than 6 months. And they're really expensive. So it's like a high throughput to look at potentially pharmacological targets. So there's a lot of cool things that are going on preclinically. And then we can play with the electrodes to measure oxygen and all kinds of other different neurotransmitters. And then they are, some of them, I think they just got the first one approved for human work. So if I don't have any more questions, thank you all so much for your interest and your time.